animals. Now's the time to catch up, okay? All right, so in your preparation to sort of switch your mind to thinking about something to do with biology, I want you to watch this little video. And it's going to last for about a minute or so. Right? Hopefully your mind is transitioning to thinking about things that have to do with science. I'm looking at these little furry guys. Chase, awesome. Playing with the cat, it's chasing something, right? Obviously a cat is a pretty sophisticated animal. We can have a debate about what's more sophisticated, dogs or cats, but nonetheless, you know, they're pretty sophisticated. People even say you can train them. Well, what if you could do the same kind of thing? You could play fetch, as it were, with a laser pointer game. Not with a cat, not with a multicellular organism, not with an animal, but you could do it with just one cell. Like, is that possible? And I'm going to show you this video. This is one cell. This is an amoeba called Dictyostelium discoideum. People who work on it just call it Dicty for short. And what you can see kind of in fuzzy, it's kind of got like a, I'll point it out to you, this thing right here. That's a pipette. So this is what you would see looking under the microscope if you could watch Dicty. And look what they're doing. They're kind of teasing it, right? Let me play it again real quick. They're teasing the poor little cell. They're putting this little molecule out on the dish that this cell absolutely loves. And just at the moment where it almost gets to where that chemical is coming out, they move where the chemical is coming from. They move the pipette. Very similar to what you just saw for the cats. So what's going on here? This unicellular organism doesn't have eyes, it doesn't have a brain, it's just one cell. It doesn't have neurons, right? But it's interpreting something that's coming from the outside. And that's what cell signaling is really all about. Yes, it is communication between cells, we'll talk about that. But it's the way that cells can interpret what's going on outside and do something about it internally. So for this little single-cell organism, it loves that chemical and it's going to go in the direction of where the chemical is coming from. Okay, so the questions to be answered in today's lecture, I'll run through them quickly. Again, this is more for your benefit, things to kind of what to watch for as we go through the slides. So there's a few here we want to talk about that. How do cells sense their environment? How can a cell that's in your pancreas, an internal organ in your abdomen, tell your triceps, this muscle, what to do? That's kind of crazy. How can they talk to each other? I'm going to link back to some ideas that we've already talked about. Talked a lot about weak forces. They're going to come up again in this lecture. What can they do? We talked about how important they are for stabilizing protein structure, but what else? Why are these weak forces so important? And then can a protein ever have more than one function? We thought about some functions of proteins the last time I talked to you, but is it possible that they could actually do more than one thing? The theme here for these two lectures, today and on Friday, is all about communication. That's what cell signaling really is about. Interpreting the environment, relaying that information either internally or to another cell. It really is about communication. So I'm going to put one more sort of organizational slide in here for you so that you can refer to it as kind of a roadmap for the next couple of lectures that I'm going to give. So I'm going to introduce signaling to you, big picture first. Before we get into the nitty-gritty of which protein interacts with which protein, we'll do that, don't worry. But before that, let's get a big picture. Let's get an overview. So we'll talk about some of the basics, and I'm going to highlight some really fundamental concepts that are going to be important throughout signaling and in fact throughout our course. I'm going to talk to you about signaling organized by receptors. And if you don't know what a receptor is, for right now, I just want you to picture an antenna, like you might see on the top of a building. That'll come back in a moment. But I'm going to organize signaling according to receptor. And of course, I'm going to show you examples of actual real-life signaling pathways that control physiological processes, okay, so that you can kind of tap into something that you might know or at least be curious about. And then we'll end with, well, how do you shut these communication pathways off? It's one thing to get them going, but when you're done communicating, how do you shut them off? So that's going to be the last thing we talk about. So perceiving and reacting to the outside world. That's what the Dicty was doing. And that's not a foreign concept. And if you look at these images of these animals, you can probably pick out what they're using to react to the outside world. Look at the picture of the mouse. It's got these really elaborate whiskers that it uses to kind of tell its way, around in the dark. Look at the bat. What is a bat using? Just real quick. I know I'm getting into organismal biology, but what are bats using? Their primary means of sort of, perceiving the outside world. Anybody know? Is it their eyes? No. What is it? Yeah, somebody points to their ears. It's sound, right? They're using ultrasound, right? This guy, the owl, uses its incredible eyesight. And then you have this octopus, which is really kind of incredible. Apparently, there's very elaborate neural networks here, even in every single one of the tentacles of this animal that let it perceive the outside world in really complex ways. That's great, but we're talking about cells right now in this course. You'll get to organisms later, but which of all the proteins that are in the cell could actually play a role in relaying information? I want you to look at this cartoon. This was actually a slide that was in my proteins lecture that I wanted to kind of end with. I showed you how proteins come to have their shape and all that stuff, but I said there's the workhorses of the cell. They're doing the job, many jobs inside the cell. So like what, for example? So this was meant to give you some idea of what can proteins do inside the cell. And I've named for you several different major classes of proteins in the cell. Just look at this for a moment. Can somebody just, which of all of them that I listed do you think might play a role in relaying information from the outside world into the cell? You can pick anything there. Yeah, please. Go ahead. Receptors. I mean, so that's an obvious one. Why did you like receptors of all of them that I showed you? Can you explain, your rationale?
Well, because it, kind of what you said, right? It helps take, what it helps take on and have it, have it process it, but it's
inside the cell, and everything that's in this pinkish purple, that's inside. So the major way that a cell is interpreting it, that it can tell what's out here, is through proteins like this, a receptor, where part of it is actually sticking out into the outside world, and the other part sticks inside the cell. Now, to be fair, I can make an argument that any one of the cell categories that I listed here actually do play a role, ultimately, in relaying information. These guys, well, I said signaling proteins, so they must have something to do with it. I didn't get more specific. I will later. But in fact, any of these really could be said to play a role. But receptors was a good choice. That's especially thinking about the analogy to antenna. That's a really good choice. So we're going to spend a lot of time thinking about receptors, because there's several major classes of receptors. But I want you to see, again, let's go with the kind of antenna analogy for a moment. When we talk about these receptors that are uniquely positioned to where a portion of them sits outside the cell, I want you to think about that class of receptors just for a moment. I'm going to introduce some terms to you that apply to receptors, but really they apply to many different types of what we call integral membrane proteins or transmembrane proteins. That's something that Dr. Pavlonis mentioned to you the other day. So we're zoomed in here on just one part of the cell. This thick purple line is the plasma membrane. In blue, we would call it the extracellular space. It's just the stuff outside the cell. And what I want you to see with this receptor is that one huge domain is this part here. The part that sticks out into the extracellular space is called the extracellular domain. But there's two other important domains. This domain actually crosses the plasma membrane. Part of this protein is embedded in the lipid bilayer. That's really important. And then the other major domain of a receptor is this one. It's the one that sticks inside the cell. It's called the intracellular domain. It's in the cytosol. It's exposed to the inside of the cell. So can you see how in this one molecule you're bridging outside world to what happens inside? Just one protein is capable of doing that. And those terms, intracellular, extracellular domain, transmembrane domain, they're important for receptors, but other types of transmembrane proteins too. Now I'm going to show you this image, and you might be like, oh boy, here we go, right? He said he's going to teach us about signaling, and I've always heard that it's complicated, and I'm not wrong. You aren't wrong. It is complicated, absolutely. But I'm going to show you it doesn't have to be. You can actually break it down into some pretty easy component parts. But I do want you to just take a moment. I'm having, of course, I'm older than you, and I wear glasses. I'm having trouble seeing what those words say. I can tell this one and some others, but there's a lot going on there that's hard for me to interpret. But does it look kind of like a map? If you've ever seen maybe a map of a subway system in New York City or maybe Washington's Metro, something like that, it kind of has that look to it, doesn't it? And if it were just a map, if it were just a subway map, you'd be looking for tracks, you'd be looking for stops, right? And that's not a bad way to think of it. Like if you thought about, well, if I'm going to move from here to down here, if I want to go around that route, I got to stop, at this stop, and I take this track to this stop, and I move to another stop, etc. And so if you start to think about a diagram like this as being a series of steps, kind of like how would you get from point A to point B, that's not a bad way of thinking about it. And of course, it's a little bit different from that, but that's okay. So what can you notice in this huge image of actually lots of signaling pathways going on all at once? You can break this down a little bit. We just talked about receptors. You can see some receptors here, and they're the proteins that are out here. This is the plasma membrane, and so you can actually see both of the leaflets of the bilayer are drawn here. So like this orange one and all three blue proteins up here, these are all receptors, but they're different. And even if you just looked at their shape, the way they're drawn, you would say, yeah, there must be something different about those receptors. But one thing you can tell is this one cell actually has lots of different receptors on its surface. That's a good thing to know. As I said, there's many different sequential processes happening all at once here. And I want you to, that word sequential, like one thing leads to the next, which leads to the next, that's really key when we talk about signaling pathways. And when you look at the diagram, you might be able to see that there's two different kinds of symbols used, arrows predominantly, and they're in black. And what I want you to appreciate is that when you see that arrow, let's say between protein A and protein B, that means activation. So protein A in some way activates protein B. If the symbol between them is an arrow, that's what it means. The opposite of that would be, in the cartoon, it's shown in red. If you see this little thing that looks like a sideways T, that stands for inhibition or blocking. So in this case, protein A would be blocking whatever it is protein B does. That's just shorthand that we use all the time in these signaling pathway diagrams, just to show how one protein can affect the next one in line in the sequential process. Well, one thing that makes this cartoon very, very complicated is that you're seeing every single step between what gets the process started, which involves the receptor, and all the way down here, which is, well, who cares? Like, if you do start the process, what is the end point? So I'm going to refer to all those steps in between as intermediate steps. But in the end, what we're doing, what signaling is all about, is changing cellular, I'm going to use the word behavior, right? I know that's kind of humanizing the cell, but behavior. Something is changing inside the cell. A process is changing because of signaling. That's why signaling matters. It changes something that the cell is doing. So I want you to look very quickly. You don't have to look at the intermediate steps at all, but I want you to answer this question. What is the output here? With all the signaling steps that I'm showing in this cartoon, what happens in the end? If a cell receives the signals that are being shown up here on the top of the diagram, so what? What happens to the cell? Even if you don't know what the term means, can you just kind of follow the arrows to the ultimate endpoint? Yeah. Yes, you're absolutely right. If you follow the arrows all the way down, all roads seem to lead to this thing right here that says apoptosis. Has anyone ever heard of this term before in biology? Some of you have. For some of you who's raising your hand, I'm going to pick on you. Tell us, like in just layman's terms, what does apoptosis mean? Yeah, that's right. It's cell suicide. So in response to these certain signals, this cell is going to kill itself. That's the output of the signaling pathway. Here's what it looks like. This is one type of microscopy that gives you very three-dimensional type images. This is what one normal type of cell, these are two cells, this is what they would look like, rather spherical. There's some texture to their surface. They're sending out these little projections, but if you had to say the general shape, it would look like a sphere. Look at the cell that's undergoing apoptosis. Something very different is happening here. It looks almost blistered. It looks like there are these little pods that have all accumulated on the surface of the cell. Something is going on with that cell. And that's a hallmark of apoptosis, to sort of make these bubbles essentially on the surface called blebs that are eventually going to be eaten up by the immune system, cells of the immune system. So one example of an output of signaling that I'm telling you about is a cell could kill itself. So I said I wanted to give you an overview. There's an overview. Now you kind of see what signaling is all about. But I'm going to break this down and to tell you some things that are in common to any type of cell signaling pathway that you might study. You're going to find these same components almost with no exceptions. At the top and bottom, these things you're okay with. There's some sort of input, right? For the dictyostelium, the input was that chemical that it loves. The output was it moved. A moment ago, the input was a signal from another cell. The output was the cell killed itself. The intermediate steps are all here. And you won't be surprised to see that at the top of the list is the receptor. Right, the thing that's sensing what's going on, the thing that's receiving the signal. I'm going to put this is a major, I'm really simplifying things by saying transducers. There are going to be a lot of transducers in there, as you saw in that diagram, but okay, they're propagating the signal. At the bottom is some protein that you want to change the behavior of. Maybe you want to activate it. Maybe you want to change where it is inside of the cell. That's an effector or a target. That target protein is going to give you the output. Maybe it makes the cell die. Maybe it makes the cell move around a petri or something like that, okay? Now, I said a moment ago that those arrows mean this is activation. So if I say input goes to receptor goes to transducer, well, the receptors must be activating the transducers. There's an arrow there, right? But a moment ago, I said there's another type of symbol you should be looking for, which is this, inhibition. So cells tightly control signaling, right? And they almost always have the option of tweaking it a little bit, turning it up, turning it down, or even turning it off. And so there can be inhibitors like this one or this one that could affect different steps of the pathway. And there's my little sideways T, the inhibitory signal that I showed you. Yeah, there could be inhibitors inside cells, but if you're a pharmaceutical company, you might make one of these in the form of a drug. If you want to turn off the signaling pathway, you find one of these components that's a good target, and you make a small molecule, a little organic molecule, that goes and binds to it and makes it stop. Okay? All right, I'm going to show you another signaling pathway, just to kind of put this into practice a little bit. So let me explain. This is a really quick, rudimentary cartoon of a cell. So this black line is the plasma membrane. Anything on the top of this cartoon is outside the cell. Anything below the arc of the plasma membrane is inside the cell, and that little half circle represents the nucleus, or the nuclear envelope. So what am I showing you here? Well, I want you to look at this for a minute. I want you to tell me two things. What's the input? And then what's the output? So I don't need you to look at intermediates right now. I just need you to look at what starts it and what ends it. So who can give me the input for this pathway? What happens? What gets the whole thing started? Anybody have an idea? Yes? So you're looking up here. Yeah, you were right to look. Let's look at the receptor. We said that's up top, right? One of the first things that happened, and you kind of surmised that kind of hourglass-looking thing was a protein. You're absolutely right. What it says is that protein is coming from a pathogen. This could be a virus or a bacteria that is out there and potentially infecting an organism. Okay. So pathogen recognition, a protein from a pathogen gets us started. What's the output? What's the so what here? So you started the signaling pathway. And what difference does it make? What does the cell do in the end? There's a lot of intermediate steps there, but in the end, what happens? What does it do? Your head, yeah. Yeah, it's spitting out. Okay, that's what secretion is, just the cell spitting stuff out. It's spitting out these molecules that we call immune effectors that can go and kill that pathogen that started the whole thing off. This is immunity at one of the most basic levels. Your epithelial cells that are in the airways in your gut and your reproductive tract, that's how they work. Okay? They're looking for invaders, and when they find one, that's what they do. Of course, we have much more sophisticated aspects of immunity in vertebrates and especially humans, but this is a rudimentary immune signaling pathway. You just figured something out about physiology from understanding the underlying cell biology, the cell signaling, okay? Okay, let's go back to just zoom in on a piece of that signaling pathway that I told you about. And so maybe one thing that's becoming apparent here, as we talk about what are the very beginning steps of a signaling pathway, is very frequently that a common input to a signaling pathway, to get the whole thing started, is some sort of extracellular molecule that binds to a receptor. That I think you're okay with. One of these got cut off, but there's actually a third receptor up here, but I'll just point these out to you. You do not need to know these names. You do not need to know these names. I'm just showing you these as examples of what gets the whole thing started. This molecule right here called FAS, L stands for ligand, which I'm going to come back to that term in a minute. That binds to this receptor called FAS. A different one called TNF alpha, a different extracellular molecule, binds to this receptor called the TNF receptor. And if I could show you over here, there'd be yet another extracellular molecule that was binding to the growth factor receptor. So different receptors are binding to different extracellular molecules. That initial input is followed by what we're going to call downstream steps. So upstream would be the very first things that happen in the signaling pathway. The receptor binds to this extracellular molecule that we call a ligand. After that are what we call downstream steps. These are all these very, we said very complicated kind of intermediate steps that are happening. So I already said that activation or inhibition of other proteins, that's really what's going on here. What I hope you can see is that one step leads to another. I already said that it was sequential, but I want you to start thinking about signaling pathways as step 1 leads to step 2 leads to step 3. So let's look at an example. I highlighted here for you just one example of what I'm going to refer to as a cascade. Cascade, if you thought about waterfalls, right? A waterfall may come down a cliff face and the water accumulates in a pool, and then maybe goes into another waterfall, and that goes into a pool, so that's where that word cascade comes from, okay? And in this case, just look at one cascade. In this case, this little molecule called TRIF gets activated, and it activates another molecule, another protein called RIP, and that one activates another protein called JNK or June kinase, and that activates, again, names aren't important, but what I want you to see is one thing leads to the next, leads to the next. That's a cascade. We often see that in signaling. This is an important point that I think just zooming in on this one part of the signaling pathway illustrates really well. I want you to focus for a moment on this protein right here called procaspase A. Look at how many arrows point to procaspase A. If we go back to our subway map, this would be like a hub. This might be a place where a bunch of lines feed into it, and then maybe you have to take another drain from there, right? So when we see this, when we see that there's actually many different pathways, I'm going to highlight them for you in a moment. In turquoise, what I did is highlighted, look at all the pathways that all seem to converge on this one protein called caspase 8. This is an example of something that we call crosstalk. When multiple pathways all seem to converge on one point or on one protein, that's crosstalk. Even though those signaling pathways started out with different inputs, they involve different ligands, different receptors, one of the downstream steps is in common. So that we call crosstalk. There's many other examples of crosstalk, but I thought that was a good one. Let me stop there for a minute before I go on to something else. Are we okay so far? Can I answer any questions before we move on? Good? All right. So I'm going to switch to the so what a little bit, right? I gave you one example of output could be the cell kills itself, but that can't happen very often or you wouldn't be alive, right? Certain cells have to kill themselves, but obviously we should expect different outputs. So let's give you some examples of why does signaling matter? What are we controlling? When I said that the behavior of the cell changes because of signaling, like what? Well, this goes back to something we just heard about from Dr. Pavlonis' lecture where she talked about ion channels, and certain ion channels can be regulated, whether they allow ions to cross the plasma membrane or not, maybe in response to some external cue, some external molecule that's coming from the outside of the cell that binds to it. It would bind to the extracellular domain of this channel, and when that happens, it allows the channel to open up and allow some ions in. These could be potassium ions, could be sodium ions, etc. So ions crossing the membrane might be an output, and that's usually kind of a very short-term, a very quick response. It doesn't last very long, right? But it can be very important for things like action potentials in neurons. What's another example of an output? You might activate an enzyme. Let's imagine that you're a bacterial cell, and you all of a sudden, one of your little receptors tells you, hey, there's glucose out here. Woohoo! Energy. What it needs to do is activate the enzyme that can break down that glucose and give it ATP, right? The energy currency of the cell. Maybe that enzyme that breaks down glucose was dormant, right? Or inactive. Maybe it wasn't active until a receptor said, yes, glucose is present in the environment. So an enzyme becoming activated, right? This is just the general idea of how enzymes work. We'll come back to this later, but the enzyme, because of a signal coming from the outside, was activated, and it made its product. Maybe it gives you a precursor to ATP in my example. This is another really important one, and it happens to be the output of so many different signaling pathways. You almost get tired of hearing it when you do study signaling pathways in detail, but it's a change in gene expression. Now, that term alone may not mean anything to you. Could somebody rephrase that? If you were going to put a change in gene expression in terms that like a non-scientist can understand, can you use different words to say that same thing? What does it mean for a gene to be expressed or not? Yeah. So you said changes the blueprint. When I hear blueprint, I think DNA, right? So we're actually a level above that. So, okay. Okay. We can solve that problem. Okay, so one way that you might say a change in gene expression to somebody who's not a scientist is just, is the gene turned on or off? Is the gene turned on or off? Are we making mRNA any better? Are we making mRNA, and is that mRNA becoming protein? Right, that's really important, and a lot of times cells are making that decision. Do I turn a gene on or off based on signaling, okay? Those can be more long-lasting, because that change in gene expression may last for a little while, certainly longer than a channel would open, okay? Okay, so we've thought about kind of some basics of signaling. What does it look like? What are we looking for in terms of activation, inhibition, things like that. We've thought about outputs. On the big picture, there are kind of three, or more than that, I guess, several different categories of signaling that you should be aware of. Now, in any of these examples, one thing that you'll see is in common is that you're always going to find that there is a cell that is sending the message that we'll refer to as the signaling cell, and another cell that is receiving that message that we're going to call the target cell. I just put above the arrow information. Okay? So they're relaying information to one another. Remember, cell signaling, yes, it's about interpreting the outside world. It's also about how cells talk to each other. So what I need you to understand is that cells can send messages over short distances or long distances, and I really mean like physical proximity here. How close are the cells to one another? Are they direct neighbors where they're literally touching one another, or is this the example of the pancreas and the tricep, right, which are whatever meters apart from each other if you're a tall person? Okay. So one way of categorizing signaling is about these different types, the distance really between the cells that are undergoing or involved in signaling. So signals sent from nearby cells, they represent one category, and there's three different types. So we're talking about short distance signaling here. One is something called juxtacrine signaling. This happens when the signaling cell, called the secreting cell here, and the target cell are literally touching each other. In the figure it says in contact. They are literally touching each other. They are neighbors in a tissue. But let's say that the target cell is a short distance away from that signaling cell. They're not touching each other, but they're not too far apart from each other. Well, then we're doing something called paracrine signaling. So the target cell that has receptors for the signal is nearby the cell that is letting out or secreting these signaling molecules. Paracrine signaling, short distance away. And then the last one is, it's a way of the cell talking to itself. And what I mean is, the signaling cell and the target cell are one and the same. So look at this. We call this autocrine signaling. That's easy to remember. We think auto is meaning self, right? So in autocrine signaling, the same cell that is releasing the signaling molecule has a receptor for that very same signaling molecule. That's autocrine signaling. So these are all examples of short distance, the target cell, signaling cell are very close to one another. Well, I said there can also be long distance signaling. And this is a case where, as I said, maybe there's a cell, I don't know, maybe it's a neuron in your spine that is sending a signal to a muscle or something like that. I gave you the example of the pancreas a moment ago talking to muscle. So in this case, endocrine signaling, you want to think of it as long distance. That's one thing to remember. It's going to involve the signaling molecule traveling through the blood in most cases. And more often than not, what that signaling molecule is a hormone. And insulin would be an example of a hormone that works in this way. So if you want to regulate whether or not a cell takes up glucose from the blood, you do that through insulin signaling. And that's endocrine signaling, using hormones that travel through the blood to go a long distance away. So that cell that makes insulin there in your pancreas is a long distance from your tricep. But if it puts that insulin molecule into the blood, now the blood travels to the tricep, and the tricep has receptors for it, right? That can change its metabolism as a result. Okay, so I gave you the analogy a moment ago about receptors, and I said that I'm really going to organize different types of signaling based on the receptor, and I think that's a key thing to remember, is that receptors are really kind of, yes, they're tuned for certain types of input, just as you might see the antenna on the top of a building. You could have antenna for cellular signal, for radio signal, for microwaves, or whatever else, right? There can be lots of different antenna out there. And I want you to think that it's the same way in cells. I showed you in that initial very complicated image that one cell had several different receptors on its surface. So here's an example of 3 major classes of cell surface receptors. I'm going to bring them all up for you at once. So 3 different types of cell surface receptors. The first one is an ion channel receptor. An ion channel receptor, that's the first class. The second is something called a G-protein coupled receptor. It's this one that kind of snakes across the plasma membrane several times. And then the last category is one that I'm going to call it enzyme coupled receptors. Enzyme coupled receptors. And we're going to go through each of these individually, and as we do so, we're going to illustrate some things that happen when these receptors are activated. We want to know, how do they work? What is downstream of that? In the case of the cell surface receptors, all of the molecules that bind to them, these signaling molecules that I said the word before, but here it is in purple, ligand, okay? That is what we call the signaling protein that binds to the receptor. Ligands bind to receptors in the same way that an enzyme binds to a substrate. Think of it that way. So in the case of cell surface receptors, all the ligands are out here in the extracellular space. In the cartoon, what's in white is extracellular, and in light blue is inside the cell, inside the cell. These are all transmembrane proteins, meaning that part of them is actually embedded in or threaded through the lipid bilayer of the plasma membrane. And some of these receptors, as you'll see, have helper proteins. Some of them, in order to do their job, need additional proteins that are tightly associated with them here in the cytosol. That's the case for these guys, the G-protein coupled receptors, and some of these, the enzyme coupled receptors. So we're starting to think about different classes of receptors, and I gave you 3 that are sitting there in the plasma membrane. Now stay with me, because I'm going to shift it on you. There is one more class of receptor that breaks this rule, that isn't on the surface of the cell. It's actually inside, okay? This is one more class of receptor. And the thing about it is that if you think of it as a different type of antenna, well, it must be picking up a different type of signal. And I'm going to show you an example of the type of signal. It's here. Here's an example of a type of signal whose antenna, whose receptor is actually inside the cell. And the signaling molecule, or this ligand, is called cortisol. Stare at cortisol for a second. Look at it. I'm going to ask you a question about it. Cortisol, okay? I hope you had a chance to take a look at it. Let's answer a question about it. Based on that structure, you can go back if you want in the slides. Based on what you can tell from its structure, what can you conclude about cortisol? There's a few different choices there for you, okay? And this goes back to some lectures that we've already had, so we're going to kind of synthesize some information from previous lectures. What can you tell about cortisol? I'm going to stop you. This is good. It seems that like 2/3 of us feel that cortisol is a polar molecule. Let's look at its structure. For those people that put that as their response, why? Why do you think it's polar? What about it tells you that it's polar? Yeah. Ooh, I like half of what you said. So if you didn't hear, he said, it looks a lot like cholesterol, which is a polar molecule. I mean, I like that. I like that, okay? This is good. If you recognize that that looks like cholesterol, that's fantastic, because it does look a lot like cholesterol. There's cholesterol right there. Very, very similar. If you said that it was polar, maybe you said that because of these things, these little hydroxyl groups here, that might be making you think that it's polar. You're not wrong, they are polar functional groups, but overall, because this molecule has these 4 carbon rings that are all associated with each other, it actually counts as a non-polar overall molecule. So actually, in the questions that I asked you, you might have said, well, it's non-polar, and then the other thing I asked is, can it get across the plasma membrane? Well, if I put cholesterol right up against, right side by side cortisol, this might influence your answer. Where do we find cholesterol in cells? Talked about this in the membrane lecture. Where do we tend to find cortisol? Dr. Jackman told you, I believe. Where do you find it? Is it in the cytosol? Is it floating around in the cytosol? I see people saying, no, that's right. Where else would we find it? Only a few more choices, right? Think of that cartoon of the cell I showed you. If it's not in the cytosol, then maybe a membrane? Maybe the, yeah, it's right there in the lipid bilayer of the plasma membrane, right? Well, if this thing can mix with lipids, then so can this, right? And so as it turns out, cortisol counts as a ligand that can enter the cell without any help. It doesn't need a transporter. So Dr. Pavlonis told you about this, that certain molecules, if you're a big molecule, if you're a polar molecule, you need some help getting across the plasma membrane. But others of them can just pass right through. We think about like oxygen or something like that, right? Well, cortisol is an example of something that doesn't need any help. It can pass right through. And so the receptors for these molecules, for cortisol and molecules like it, they can pass right through the plasma membrane. So you're looking at the plasma membrane is in orange, outside the cell in blue, the cytosol is all in orange. Here's the nucleus. Here's cortisol. It passes right through the lipid bilayer because it's soluble in these lipids that make up the plasma membrane. And it's in here, in the cytosol, is where it meets its receptor. So the cortisol receptor is an example of a receptor that you find not here on the plasma membrane, it's actually inside the cell. It's an example of something that we call a nuclear hormone receptor. So it's binding to cortisol that comes into the cell. Watch what happens to the cortisol receptor. After it binds to cortisol in blue, look what happens to it. It's allowed to go inside of the nucleus, and it sits down on DNA, and it's affecting gene expression, it's affecting transcription. Remember, that's a very common output. So in response to cortisol, which is a stress hormone, right? That's why you would have cortisone floating around in your blood. You've been stressed in some way. Well, certain genes that can respond to that stress need to be turned on. And in this case, this is an example of a type of receptor that breaks the rules of having all your antenna on the plasma membrane. Well, you can actually have some inside, okay? I'm going to take a little diversion here for 1 second because what you're seeing, ligand-receptor interaction, is so important. And in fact, protein-protein interaction and all the signaling is so important that I'm going to take 2 slides to go off on a little tangent about that. Just because this, I mean, not just in this course, but binding, understanding binding is really important. And it comes up so often in signaling. I'm going to spend 2 slides on it here. Okay? When you picture binding, this is what you might picture. That there are 2 molecules whose shape is complementary, where this one kind of forms these invaginations or folds, this one protrudes. And when you put them together, they fit together like lock and key. Shape complementarity, like that helps in protein binding. Often we see that. But the other thing that I want you to understand is that binding is mediated by interactions between amino acids, and particularly between the side groups. And I promised you that I was going to show you another example of where weak forces are important in biology. This is it. Yes, they help these proteins to keep their shape, but if this green protein and this orange protein are going to bind together, what's going to keep them together are weak forces. And I'm showing you some specific examples of weak forces here. And the same ones keep coming up, right? It could be hydrophobic interaction, right, where you have these nonpolar groups that cluster together. They could be electrostatic interactions, where maybe a negatively charged amino acid side group interacts with a positively charged one. Or you could have an interaction like this between serines side chain and asparagine side chain, where you get a hydrogen bond. All of those count as weak forces. And the more of those that you can form between the 2 proteins, the tighter they stick to one another, the tighter that binding is. And we refer to that as affinity. What is one protein's affinity for another? How much does it like another protein? How tightly does it grab on and hold? The next slide is busy, I will promise you that, but the concept is really easy. But I'm going to show it to you for 2 reasons. One, because I think it's important that you understand something about binding that if you're going to go into medical school, you should probably know this. And 2, I want you to be okay with numbers. And I'm going to put a number to affinity. You should be able to measure the strength of the binding, the strength of the affinity, right? It could be low, it could be high. I'm going to spend 1 slide to show you that. There's a lot on here. It is not complicated, I promise. All we're talking about is 2 proteins that like each other. They're going to bind. I have my green protein in A, and my B protein is green. They're going to bind. They're going to form what we call a protein complex, the AB complex. So let's think about 2 scenarios. What if protein A has high affinity for B? Well, then more often than not, I'm going to find protein A bound to protein B. I'm going to find more of this protein complex than I will free protein A or free protein B. Is everybody okay with that? What if they don't like each other very much? What if the affinity is low? Then the opposite, right? More often, I'm going to find these 2 proteins hanging out by themselves, and only rarely, only on occasion, will I find the AB complex. Affinity is low. They're not attracted to each other very much, right? Okay, you can quantify this in something called a dissociation constant. And it's easy. It's just a ratio. That's all it is. So look at what the ratio is. If you don't know these brackets, it's shorthand for concentration, like you would hear about concentration in your chemistry course. So look at what you're doing. You're just saying, I'm going to take a ratio of the concentration of the protein AB complex is going to be in the denominator, and the concentration of the free proteins A and B, I'm going to multiply those together, and that's my numerator. So that together is called a dissociation constant, or K with a little d. Here's the thing. Like any fraction, like any proportion, the bigger the bottom number is, the smaller the fraction is, right? Just like 1/8 is smaller than 1/2. Everybody's okay with that, right? So if I'm seeing more of this, if A and B are frequently bound together and they bind tightly, I almost never see A and B by themselves. If they can get together, they stay together. That number should be high, the KD should be low. On the other hand, if it's this, where, yeah, they might bump into each other in the cell, but they're not going to stick together. More frequently, I'm going to find them apart. Well, then these 2 numbers, the A and B concentration, are going to be higher than the concentration of the complex. That makes the fraction a bigger number. Okay? Here's a table if you just kind of want to go through that and prove it to yourself. You can plug some actual numbers in there later, okay? But let's see if you are okay with this. Here's another question for you. I'm going to give you some KD values. And this might test a little bit of chemistry knowledge, because there's annotation here that you should know, like little m, big M, or little n, big M. So I'm talking about millimolar, or nanomolar, or picomolar. So look at those KD values. Which one indicates? Okay, this one, over half of you think it's 0.5 molar. That's a pretty big number, right? This is nanomolar. That's 10 to the minus 9th molar. Millimolar. That's 10 to the minus 3rd. Or pico, 10 to the minus 12, right? So those are all really small numbers. This is a huge number in comparison, meaning that that top number, the numerator, the free A and B concentrations were super high. right? So they don't like each other very much, and that's going to give you a high KD value. Their affinity is low for each other, so you're absolutely right if you picked the top value. So this comes up when we think about enzymes and substrates, but it also comes up when we think about receptors and ligands. And I put them side by side here because next week in lab, even though we're thinking about signaling today, we'll think about enzymes and substrates, and binding is still important. So you can think about them as being analogous. So in general, there's 3 things I want you to know about binding. It tends to be specific, meaning that certain proteins only bind to certain others, and they're very selective, I guess, is the way to think about it. It's non-covalent. I didn't say anything about covalent bonds sharing an electron. It's all weak forces. And it's reversible. It can be broken. This was true with proteins, right? Weak forces in proteins. We could break those through heat or pH or denaturing agents, right? Same is true of binding. You can easily break the association, particularly between binding where the affinity between the 2 proteins is very, very low. In the last minute that I have with you, I'm going to go through the first type of receptor, because there's only one slide on it, where there's lots more to say about G-protein coupled receptors and enzyme coupled receptors. So let's do ion channel receptors real quick. And this is easy because you just heard about this from Dr. Povolonis. So we're back to the plasma membrane now. This is an antenna that's sitting there, some of it is exposed to the outside. And when I said, can a protein have 2 different functions? Here's an example of where it can. Because look at this. Just the name of this thing tells you it can do 2 different things. It's an ion channel, and it's also a receptor. So look, here's the plasma membrane, here's an ion